You've annotated your genome. You knew the taxon of it if you ran taxonomic classification. You certainly could figure it out if you did similar genome finder once you had it. Even if you annotated at the highest taxon level of bacteria, it would be showing you who it was closest to. But now this is a service where, well, if I was a genome, it would tell me who I was in the greater world of genomes. Who are my relatives? Who am I closest to? The bacterial genome tree allows you to see your genome in a greater context. All phylogenetic trees do that, but this is a particularly sweet service. So the tree pipeline uses single copy global protein families. They're called PGFAMs for the data. And from these, they take the genes and the protein sequences. So it's taking both nucleotide and amino acid. Once it has that data from across the selected group of genomes, and it has these protein families, it builds an alignment of both the genes and protein family data, and it uses muscle to do this. The nucleotides are aligned by mapping to the protein alignment. Then it uses RAXML for tree generation and bootstrapping. And it also, in this particular pipeline in BV-BRC, you can have metadata. So you can see metadata on the tree, which is awesome. I guarantee you, you are going to love this. Okay, this is the publication on the protein families. The protein families are not blast generated, they're Kamer generated. So there's some noise in the alignment there, which makes them pretty good for trees, because for trees, it's looking for things that are different. So it's every genome that is annotated in BV-BRC, and before it was BV-BRC, it was Patrick and Viper and IRD, but Patrick was where we had the bacterial data. These protein families are part of every annotation. If you do just straight up annotation, comprehensive genome analysis, or metagenomic binning, protein families come with it. And these are what are used in this pipeline, and also in something like comparative systems. So both protein and DNA are used in the tree. So what it's doing is it's taking a sample of the aligned protein positions and it's submitted to RAXML for the best protein substitution. And once it finds that, and it does this other DNA model, it's using a joint analysis of both kinds of data. And then it generates a partition file for RAXML. And the alignment columns are treated as the first codon position, the second codon position, the third codon position. And for all of us who have ever had the most rudimentary biology class, we know that we're talking about genes here and also the protein. Now, there are some things to keep in mind. I've already said that the service uses the global protein families, the PGFAMs. The genomes have to share some, or it can't generate a tree. It's got to share some protein families. Now, if you are questioning whether your group of genomes have enough protein families shared between them, you could use comparative systems, and that will show you if they have at least enough protein families in common. The PGFAM selected must be single copy. Then that means one protein per genome. Now, the pipeline calls for that, but because we know we live in an imperfect world and some things don't work out the way we expect, we allow you to adjust some things. You can adjust for maximum allowed deletions or maximum allowed duplications, and we'll get into that when we submit the job. But these are protein families that aren't perfect. The service requires at least 4 genomes and allows up to 100. I can't tell you how many times people have submitted a tree with just 3 genomes and then complained to me about it. Why one wants a tree with just 3 genomes is beyond me, but sometimes we do. You're going to have to generate that someplace else. You can't do that here. Add one more. Genomes love company. All right. This is a question we often get on the trees, and bootstrapping, it's part of RaxML, and it's a method to estimate the reliability of each branch. And what the bootstrapping algorithm does, it's called resampling based support. And it tells you how confident you can be in specific parts of the tree. So it resamples the alignment data with replacement, and it's creating mini pseudo replicates. So it's building a phylogenetic tree for each replicate using maximum likelihood. And when it has all of these trees, it counts how often each clade, which is the group of genomes, appears in the replicate trees. So it's looking at this group that seems to be branching together into a group. How often do I see that every time I run these 1,000 different replicates? And so then it will assign a bootstrapping value based on how often it sees that these trees are replicated. So to interpret bootstrap values, if it's greater than 90%, that's really strong support of the clade. If it's 70 to 90%, moderate support. But you know what? You get what you get. And I don't scream when I have 70%. Well, I might add more genes to it. And if it's less than 70%, the branch isn't well supported. So what would I do? I might add in more genes to look at or get rid of some genes. This is what the input looks like, where you can choose your genomes and the parameters. And you can add in metadata, and there's a lot of metadata that you can add, and we'll go over that. Remember, all of these can be used to color the trees. Now, what does it return? A tree like this. Look, it has been colored by the metadata, the red one and the pink one and the black genome there. Those are all metadata. This viewer is really awesome. Now, the tree also gives you statistics that are important for your manuscript if you're going to publish on this tree. And the one things I always like to include are the number of genes that it was built on. I've had reviewers ask me that, like, even if I've had 1,000 genes, a reviewer has said, well, what were those genes that you use? And luckily, this service provides you with lists. So I just, as an addendum, added the 1,000 genes and said, here they are. Is that not enough for you? 1,000 genes? I also include the number of aligned amino acids, the CDS alignments, and the number of aligned nucleotides. This is data you can put directly into the publication. It also gives you ways to improve the tree. If you ask for 100 genes and it couldn't find 100 genes, it'll say, hey, if you get rid of this guy, you're going to get this many more genes. It looks like I was asking for 1,000 at that time, which is greedy. But anyhow, I like a lot of the services here, but this might be one of my favorite ones. So let's submit some jobs so I can show you just how much you're going to like.